Thanks, John, and it's a pleasure to be here. I actually had to change my topic fairly last minute, so I apologize for that, but we'll talk about overcoming resistance with BTK inhibitors. I realized that trying to talk about novel BTK inhibitors when I had very limited information would be a very short talk, which I know would be in the best interest of a talk at 7 p.m. or 6 p.m., whatever time it is, so bear with me. So I think just by way of background and putting things into context, it's really important to recognize that there are two forms of pathologic BCR receptor signaling in lymphoid malignancies. And I know that everybody knows this, but it really is important in terms of thinking about, how to place these treatments into what category. So first off, for the tonic B-cell receptor signaling pathway, that's required for B-cell survival. It's antigen independent. In contrast, the chronic active B-cell receptor pathway is antigen dependent, and it's needed to induce the germinal center response. Of note, the tonic B-cell receptor signaling pathway is independent of BTK, so it's driven by PI3 kinase, whereas the chronic active B-cell receptor signaling pathway, as you all know, is the BTK pathway is integrally involved. I think it's very important to note that different lymphoma subtypes can be independent of this chronic active B-cell receptor signaling pathway. So specifically, Burkitt's lymphoma and the GCB subtype of diffuse large B-cell lymphoma do not rely upon the chronic active pathway. This just gives a cartoon of the BTK structure, highlighting the phosphorylation sites and the multiple regulatory partners involved that at least were known in 2009. And again, putting all of this in context, thinking about the different targeted therapy paradigms, as is well known in the recurrent mutation model, such as highlighted with CML and the imatinib story, there's direct inhibition, which leads then to direct cell death. So there's a known mutation that's inhibited. In contrast, for ibrutinib, there's no known BTK mutations. And instead, it's inhibiting the B-cell receptor survival pathways and also impacting the microenvironment, leading to cell deadhesion and subsequent cell death. So in terms of ibrutinib resistance, as was just illustrated, the genomics here demonstrate minimal genomic changes. So I have less information than what was just highlighted. This was from the ASCO abstract, where 8% of the patients demonstrated some evidence of progressive disease. And of those, the 3 cases that were profiled, and again, we just heard about these, which were the point mutations of the cysteine to the serine, and then the 1 gain of function mutation, which was wild-type BTK. So how can we track resistance? Because this is going to be important in the clinical arena. We need to know how we're going to be following these patients. I think there's 3 different ways that we can look at this. I do think that the genome expression profile is still going to be important. We know about MYD88 mutations as well as CARD11 mutations in certain lymphoma subtypes. We could look at phosphoprotein expression. It's been discussed several times today, looking at resistant subclones and how that's going to impact treatment. So if there's some way of tracking that, I think it would be very important then in terms of deciding, is this somebody that needs to come off of ibrutinib, or is it somebody that we should be adding on another drug to? And then lastly, we can look at the expression of cytokines to determine the impact of the treatment with the BTK inhibitor. So an example would be with CXCR4 expression. This has been highlighted with the WIM mutations identified in patients with Waldenström's. So there's been several published studies looking at the different mutation frequencies in CLL. This is one published in the New England Journal by Wang and colleagues, and I'm showing this here just to highlight that the MYD88 mutations were present in approximately 10% of patients. So this would be something that we know we can be following. And then moving on to the potential following for subclones. This was a study that was published by Irish and colleagues, where they essentially are looking in follicular lymphoma cell lines. And they're doing, these are 3D plots of flow cytometry in B-cell receptor stimulated cell lines. So they're looking at ERK, BCL2, and CD20 expression. And you can see that if I know which one is the highlight here, that in this LPJ023 cell line, there's this subpopulation that when they look at further is not phosphorylating ERK or PAKT. So then they went and looked at patient samples and identified that within patient samples they could see a group of patients who had higher expression of this subclone. They named that subclone lymphoma negative predictive cells, so LNP cells. They found that patients that had high expression of LNP cells had worse outcomes. They went ahead then and looked at both populations, so a high expressing population and low expressing population. And at basal signaling, there is no difference in phosphorylation. However, with B cell receptor stimulation, there is a significant difference in phosphorylation, so lower level of phosphorylation for those who had high expression of these LNP cells. What I thought was very interesting here was that it was reversible. And so if they treated the samples with peroxide, the phosphorylation was overcome. So then you see similar expression once the B cell receptor stimulated cells are treated with peroxide. So again, kind of giving us evidence that perhaps this is something that we can actually overcome when we see these subpopulations that can be treated and can be expressed, the phosphorylation can be expressed again. This is data looking in mantle cell patients that were treated with ibrutinib. And this just speaks to the cytokine expression. So in mantle cell patients treated with ibrutinib, CXCR4, CD38, and Ki67 expression goes down with treatment. So again, these would be other potential means of tracking resistance in patients treated with ibrutinib. Fortunately, I just learned today that CXCR4, while I know it's highly expressed in CLL, apparently the expression is increased with ibrutinib treatment was mentioned today. So probably not the best measure of following patients with CLL treated with ibrutinib. But I think that we can continue to look for other cytokines, and then possibly Ki67 will also be a useful measure for tracking resistance. We heard today that the Ki67 in the peripheral blood is typically low. Perhaps if we start to see a climb, that would suggest somebody who's becoming resistant. Then moving on to thinking of some rational combinations to overcome resistance. Sort of in the future, I think one rational combination would be to look at a BTK inhibitor along with an IRAK4 inhibitor. There's been data suggesting that there's preferential association of phosphorylated IRAK with MYD88 mutants. And there have been some novel compounds that have been developed that do inhibit IRAK4. So hopefully these sorts of agents will be coming into the clinical arena at some point. Another rational combination, I think, would be looking at a BTK inhibitor with lenalidomide. It's known that lenalidomide induces type 1 interferon responses that can overcome these MYD88L265P mutations. So that obviously makes sense in terms of that rational combination. There already is a phase 1 study which is underway. Dan Pollier at University of Colorado is the PI for that study. And then there's already a lot of discussion today about the rationale for combining a BTK inhibitor with a BCL2 inhibitor, so I don't need to really go through that, but what was mentioned was that MCL1 and BCL-XL protein levels decreased in ibrutinib-treated patients, and we saw that the ABT199 exerted cytotoxicity at any stage of treatment with ibrutinib, so that seems to be a very rational combination as well. So what can we do to enhance efficacy? Again, I think we really need to consider the unique pathways that are involved for each lymphoma subtype, so that speaks to this whole tonic versus the chronic activated. So obviously, some of these drugs are not going to work in certain lymphoma subtypes. We need to be cognizant of the sequencing of combination regimens. So it was already discussed today, the questionable added efficacy of adding cytotoxics. So are we really gaining much with the BR combinations with ibrutinib? And I think that really remains to be seen. It was hinted to today as well by John Bird that there's maybe some possible antagonism with rituximab as well. So in this recent publication, ibrutinib blocked NK ADCC which decreased the efficacy with concurrent administration. Again, we know that rituximab works in several different ways. Whether or not this has any bearing in the clinical arena, I think remains to be seen, but I think we do need to be cognizant of it. And at least in that study, in cell line studies, what was demonstrated was that sequential administration of the rituximab and ibrutinib actually seemed to be a more effective combination than doing them simultaneously. There has been some data of another BTK inhibitor, specifically AVL-292. I think that the responses, despite having full BTK occupancy, were a little bit disappointing, and I think that led to questions as to whether or not ibrutinib may actually have some off-target effects. But I think it's important to highlight that the patient population here is very diverse. It's not CLL. There's lots of large cell lymphoma patients, and again, we don't know whether or not those were pure I don't know whether or not those were ABC patients, ABC DLBCL. With regard to novel BTK inhibitors, the ones that are currently under investigation, the covalent irreversible BTK inhibitors similar to ibrutinib or AVL-292, there's a phase 1b/2 study which is still ongoing. In addition, there's a phase 1b study of AVL-292 plus lenalidomide that's being conducted by Professor Salles in France. And then next there's ACP-196, and there's a phase 1 study underway, and it excludes patients who've had prior treatment with BTK inhibitors. The non-covalent BTK inhibitor that we're aware of is dasatinib, which is a multi-kinase inhibitor. Didn't have very impressive results. But I think in terms of thinking of what would the ideal BTK inhibitor be, in my mind, that would be one that would bind in a unique spot from ibrutinib such that we could overcome these CIS481 serine mutations. So in conclusions, I think that longer experience with ibrutinib is inevitably going to demonstrate more resistance, so I think we need to be mindful of this. I think it would be very helpful if we can find ways of actually monitoring for the resistance so that we can actually guide future therapy. And then lastly, thoughtful combinations of targeted agents with non-overlapping toxicities are anticipated to hold promise. Thanks.